Imagine a magical substance that can turn into any part of the human body. Just had a heart attack? The substance will fill the damaged tissue with brand new beating heart cells. Going blind because your eyes are degenerating? This substance can restore your sight. It sounds like something out of a science fiction novel, but scientists have discovered how to make something remarkably similar. They're called Induced Pluripotent Stem Cells, or iPS cells for short. Before we dive into how iPS cells are revolutionizing medicine and research, we have to understand what normal stem cells are and why IP stem cells are so magical. How does a single cell turn into a full human being? It all starts with a process called cell division. The fertilized egg, called a zygote, grows and divides, replicating many times to eventually make up a human embryo. Each time a cell divides, it gets closer to its final destiny as a cell. Think of it like a college student getting closer to completing their degree with every course they take. In this case, their degree might be a muscle fiber, a retina cell, or a neuron. However, human development is a bit more complicated than just growing and dividing. There's a couple of different stages when it comes to how exactly each cell and daughter cell differentiates into the different cell types that make up a completed organism. After only a few divisions, the zygote reaches the 8-cell phase. At this phase of the embryonic development, every cell, even if you isolate one of them from the rest, is able to create a whole organism from just itself. This is the property called totipotency. Then, the mass divides a few more times to form a ball-shaped structure called a blastocyst. The blastocyst is a very early stage of the human embryo, and most important for stem cell research. It's made up of 2 parts. The trophoblast surrounds the ball-shaped structure and later forms part of the placenta, a temporary organ that connects the mother and the fetus, providing oxygen and nutrients to the developing embryo. The inner cell mass is made up of a special type of cell called stem cells. Unlike other cells, stem cells can make infinite copies of themselves. Each time they divide, one of the daughter cells turns into a new cell type, while the other daughter cell remains a stem cell. Instead of getting closer to their degree, like in the earlier example, stem cells just remain the same while pumping out degree-seeking daughter cells. This self-renewing property is what makes stem cells so important. The inner cell mass of a blastocyst is made of stem cells called pluripotent stem cells. They're like the OG stem cells. All other stem cells, such as hematopoietic stem cells that make up different types of blood cells, they all come from pluripotent stem cells. While pluripotent stem cells can't give rise to a complete organism like totipotent cells, with the right signals, they can turn into any cell type, from neurons to heart cells to muscle cells. These pluripotent stem cells have all kinds of applications, from regenerating damaged tissues for patients in the clinic to providing incredible models for studying diseases in the lab. There's a problem though. Recall that pluripotent stem cells come from blastocysts, which are human embryos. Harvesting embryonic pluripotent stem cells would require lots of human embryos, which, as you can probably imagine, raises all kinds of moral concerns. Then, in 2006, Dr. Yamanaka discovered something groundbreaking. After testing many different candidate genes, he found that using a harmless virus to introduce 4 specific genes into any cell could turn it, whether it be a neuron, skin cell, or a muscle cell, back into a pluripotent stem cell. The 4 genes, now called the Yamanaka factors, were a biological factory reset button. As these cells are induced by the Yamanaka factors into becoming pluripotent stem cells, they are called induced pluripotent stem cells, or iPS cells. The discovery of iPS cells, which won Dr. Yamanaka the Nobel Prize in 2012, avoided the ethical conflicts associated with embryonic stem cell research, giving lab researchers tools to model all kinds of human cell environments with just 4 factors. However, while stem cells hold tremendous promise, there's still many obstacles surrounding their therapeutic applications. For instance, remember how stem cells have a self-renewing property and can infinitely replicate themselves? Well, if left unchecked, that valuable property can also allow them to divide uncontrollably, forming tumor-like masses in the patient. Despite these challenges, the discovery of iPS cells and the Yamanaka factors has revolutionized the field of regenerative medicine, offering hope for new treatments and a deeper understanding of human biology. As researchers continue to overcome these obstacles with discoveries such as iPS cells, the potential for stem cell therapies to transform medicine is brighter than ever.